A very good evening. I bid to all the panels and fellow audience. My name is Hazel Lasius, and today I'll be presenting my final year project entitled Antibiofilm Activities of Bacterial Isolates from the Skin Microbiome. So we will go through the introduction, the problem statement, followed by significance of study, objective, methodology, results, and finally conclusion and references. Moving on to introduction, the human skin consists of various types of microflora that live symbiotically on different locations of the human body and may play a role to maintain the skin homeostasis. In nature, bacteria usually exist and grow in a biofilm structure made of extracellular polymeric substance that enable this bacterial community to resist drug treatments and also enable them to survive in harsh environments. Some microbial species are able to produce antimicrobial or antibiofilm peptides that inhibit the growth of other competing microbes or pathogens. Antibiofilm compounds, as stated by the SOMA in 2020, are important in interfering the crucial steps in the formation of biofilm, and it is a promising adjunct treatment for microbial infections. Here, as you can see from the picture, our skin consists of various types of skin microbiota, almost every inch of our skin, be it bacteria, fungi, and even viruses. But typically in this study, I will be focusing more on bacteria. Moving on to problem statement. Biofilm is a structure formed by most bacterial species that can cause a detrimental influence in humans, such as infections caused by biofilm on medical devices. The capacity of biofilm to resist basic medical treatments, such as antibiotics, is why it is such a problem, and antibiofilm agents, on the other hand, are still in short supply presently. Hence, there is a need to find new antibiofilm agents or antibiofilm peptides to be developed into treatment for bacterial infections. So what are the significance of this study? Considering the adverse effects of biofilm in human or specifically patients that is using medical devices such as implants, the finding of this study is crucial to solve the problem. And this study will help us to better understand the biofilm-related infections. This study will add some more data to the research topic with regard to antibiofilm agents, and the knowledge obtained from this study can be used to specifically kill bacterial communities that could potentially form biofilm. This study can also lead to the finding of new antibiofilm compound that is potentially effective in eliminating the bacterial communities that form biofilm. So the research objective of my study is to screen the bacteria isolates for their ability to produce antibiofilm compounds to inhibit biofilm formation, and to identify the bacterial isolates that produces antibiofilm compounds using 16S rRNA sequence analysis. So there are three parts of methodology in this study. First is the isolation of bacteria from the skin microbiome. Second is the screening of antibiofilm compounds. And third is the identification of positive isolates by using 16S rRNA sequence analysis. Moving on to the first part, I started off with first preparing the media, MSA and Mrsa. I performed a skin swab in 4 sites of the skin, which is the forehead, the skin behind the ear, the moist area, which is the inner part of the elbow, and behind the knee. After that, I directly streaked them on MSA plate for the detection of coagulase-negative staphylococci and on Mrsa plate for the detection of lactobacillus. I do perform replicate of this to ensure that I obtain pure isolates. After obtaining the pure isolates, I culture them in deep well plates for 3 days at 37 degrees C. This is to ensure that the bacterial grows into stationary phase where they produces metabolites. After that, I filter sterilize the solution to obtain sterile conditioned medium. Moving on, for the screening of antibiofilm compounds, I first culture the test organism, which in this case, I use S. epidermidis ATCC35984 as a positive control since they are a thick biofilm former, and S. epidermidis ATCC12228 as a negative control since they are non-biofilm former. After that, the SCM was added according to the phase, which is attachment and maturation. I further incubate them. After that, the media is removed by decanting and washing with phosphate buffered saline. After that, the content was discarded and washed with PBS. After washing with PBS, the biofilm in the wells was fixed by using methanol for 5 minutes. After that, the methanol is removed and air dried. To stain the biofilm, I use crystal violet. I leave it for 15 minutes and wash it by using tap water. To redissolve the crystal violet, I use ethanol and also apply gentle shaking before proceeding to the measurement of optical density. After obtaining the optical density, I proceed to compare the percentage of biofilm inhibition with the negative control. Last part of this method is the identification of positive isolates by using 16S rRNA sequence analysis. The positive isolates that I obtain undergo DNA extraction and PCR amplification of the 16S rDNA gene. After that, PCR product cleanup is performed before the positive isolates is sent for sequencing. Moving on to the results, here I provide two pictures of the attachment plate and maturation plate. I choose these two plate because the SEM in the attachment plate well A10 shows 100% inhibition of the formation of biofilm, and for the maturation plate, well in D7 shows 88% inhibition of the formation of biofilm. So after analyzing the result, here the table shows the antibiofilm activity of skin commensal isolates against the S. epidermidis ATCC35984. So as for easier understanding, I provide you with this pie chart. So for the attachment biofilm assay, 9% of the bacterial isolate shows strong inhibition, while 3% showed medium inhibition, 12% showed weak inhibition, and lastly, 76% of the isolate showed no inhibition at all. On the other hand, for the maturation assay, 6% of the bacterial isolates showed strong inhibition, 46% of which showed medium inhibition, while 15 and 33% of the isolates showed weak and no inhibition, respectively. After the positive isolate is obtained, I proceed to PCR result. I choose two of the positive isolates since they show a high antibiofilm activity. However, the PCR amplicon, I found out it was slightly larger than 1.5 kb, which is the expected size of the 16S rRNA gene amplicon. And after analyzing the result, I concluded that this could be due to a poor gel preparation and overloading of samples. The amplified DNA has been sent for sequencing in a company in Singapore, and the results are expected to be obtained within next week. This is due to the current COVID situation, which prolonged the waiting period of obtaining the result. Once the sequence has been received, It will be filtered and used to search in NCBI gene bank database for a match to attempt to identify the bacterial species. So as a conclusion, the findings of this research prove that it added some more data to the research topic with regard to antibiofilm agent. Referring to the result, it shows that the finding of new antibiofilm compounds from the skin microbiota that are potentially effective in eliminating the bacterial communities that form biofilms are possible. Next, here are my references. With that, I have come to the end of my presentation. Thank you all for listening.